so, I have two brothers and two sisters. So we have a large family, and especially when we were young, of course, that involved a lot of chaos. And we had a lot of freedom at that time. Children now seem to have very scripted lives, and whereas we had a lot of freedom. Basically, you go out of the house in the morning, let's say in the summertime, and you come back in the evening for dinner, and nobody's expecting to see you in between. So yeah, it was a lot of fun to grow up in that kind of an environment. I would say for my career inspiration, I was very fortunate to have a high school biology teacher, Dr. Rose Nelson, and she was a PhD, so she had done research, had done postdoctoral training, and she taught us biology not as a series of sort of facts, but she would teach us about discoveries and about scientists who made discoveries, and sort of the excitement of learning something new in science. And that really excited me. She also recommended to me a summer program at a research institute where I was able to experience research firsthand. And so all those experiences really excited me and developed a passion in science that I've had ever since. And so I really owe that to her. And it's one of my regrets, that she passed away before this time, because she was a very positive person and very ebullient, joyful. So she always had a beatific smile on her face, and she would be teaching us something, and she would say, "Now I want you to remember that when you win your Nobel Prize that you learned that here." And she said this, not to me, to the class. And she would say this as a statement of fact. So I don't know how she knew, but she was really a remarkable teacher. Well, I guess, the idea of hereditary passing down of traits and how this was determined by sequences within the DNA, it's kind of a very amazing thing, right? To go from these molecules to traits of living organisms. And that really interested me. And so I was first interested in genetics, sort of basic genetics, and thought I would do, sort of basic genetic research. But while I was in college, a family that we knew had a child born with Down syndrome, and I became interested in medical genetics and decided that I would try to get training both as an MD and a PhD so that I could do both genetics research and medical genetics as a clinical specialty. Right. Well, when I was a college student, I worked in a lab in Boston, and the leader of the lab, he would always say, "Search and research." Because that's the nature of science, that there are always obstacles to be overcome experimentally, and that's a challenge. And if that isn't something that's enjoyable to you, then probably science is not a good line of work, because we're constantly faced with those kinds of obstacles and challenges. And part of the fun of the work is using our creativity to come up with solutions to those kinds of problems when they arise. Persistence is a very critical attribute, absolutely. And then the other part of it that is very helpful is to have colleagues who take orthogonal approaches to the science. So they may look at the science in a slightly different way and may provide an avenue for circumventing various obstacles. And so when we were trying to isolate the DNA sequences that coded for the subunits of the hypoxia-inducible factors, we took one approach because we thought that there was only one protein that was involved. And we took an approach that involved screening human DNA sequences in bacteriophage, and we screened millions and millions of these clones, and we got negative results. So, as the leader of the research project, it's sort of my role to decide what to do. So, we could continue doing what we were doing. That didn't seem like a very good idea. We could give up and let someone else do it. That didn't seem like a very good idea either. Or we could take a completely different approach, which was, rather than a molecular genetic approach, to take a biochemical approach and try to purify the protein through biochemistry. And this was not exactly our forte. So, we owned none of the equipment that was required to purify proteins, did not really have the expertise to do that. But fortunately, across the street at Johns Hopkins was the lab of Tom Kelly, and his lab was one of the first labs to purify a protein based on its binding to DNA. And so with the assistance of his lab, we were able to do that, to purify the protein from 100 liters of cells growing in culture, and to get just enough of the protein that we could obtain some protein sequence, and then use that protein sequence to identify the DNA sequences of what turned out to be the 2 subunits of the protein. So in fact, the approach that we had been taking would never have worked, because that approach only works if the protein has a single subunit. And because our protein turned out to have 2 different subunits, only through the purification, the biochemical purification, would we have been able to succeed. And, so again, the freedom and the creativity, these are the really fun parts of the job, that no one tells us how to do our work, and as long as we're successful, we're left alone, and we can do our thing. And so to have that freedom to, not only with regards to, problem solving, but even more fundamentally, what questions do we ask? What are the really important questions that we'd like to answer? That's left entirely up to us, and obviously that changes over time. And so we started with sort of more basic fundamental questions, and then over time have shifted to more applied applications as the project has evolved. And so that's been very satisfying over the course of 30 years to be able to connect the dots to very molecular, fundamental questions. How does a cell sense the oxygen concentration and respond to changes, all the way to, can we develop new treatments for anemia, for cancer, for cardiovascular disease? As someone trained both in research and medicine, for me, I've always felt it was my role to bridge between the basic science and the clinical translation. And so, it's been very satisfying to reach that point where we see these translations occurring now as a result of these basic discoveries that were made 25 years ago. That's a good question. Well, I guess I doubt myself often. I often doubt, am I taking the best approach? Am I running the lab most efficiently? Am I mentoring my students well enough? Could I be doing a better job at that? I guess I have those kind of doubts or self-criticism on a fairly regular basis. No, well, I think the word is dumbfounded. So, it was, of course, in the US, it was the middle of the night. And I was sleeping very soundly, and the phone rang. And, when I finally was awoken out of the sleep and got to the phone in the hallway, it had stopped ringing. So, again, I was not even really awake yet. And I figured, well, maybe this is somebody's idea of a bad joke, because I didn't know what day it was. So I went back to sleep, and actually quite a while later, the phone rang again. I said, perhaps I better be quicker this time. So I got to the phone, and Thomas Perlman apologized for waking me up, and then gave the good news. And I was sort of on the phone and, my jaw just dropped and I didn't really, words weren't coming out. And of course, my wife was there and she could hear what was being said on the phone and her jaw dropped and she couldn't say much either. So we were just kind of mute. It was a very one-sided conversation. Well, everybody appreciates the importance of oxygen, right? I mean, you just have to hold your breath and you'll start feeling uncomfortable very soon, right? Because you have 100 trillion cells in your body and they all need oxygen on a continuous basis. Principally to make energy, which allows us to, for this complex organism to function, right? And the body uses glucose and oxygen in order to generate energy. And so there has to, but by the same token, oxygen at too high a levels causes damage to cells. So there has to be a very tight coordination between supply and demand. So the amount of oxygen that each cell requires has to be matched by the delivery of oxygen, which is of course carried by red blood cells through the blood vessels to each cell. And so there's a beautiful, physiological system that maintains the balance between consumption and delivery of oxygen in every one of the 100 trillion cells in your body under normal, healthy conditions. And of course, that balance is disturbed by many common diseases, including cardiovascular disease and cancer. Yeah, so, the first translation of the discovery of this system to the clinic is the development of new drugs for the treatment of anemia. So we started by studying the hormone that controls red blood cell production called erythropoietin or EPO. Patients who have a chronic kidney disease, the kidneys stop making EPO and they become anemic. And the discovery of EPO was a revolution because it enabled the patients to receive a recombinant EPO protein as an injection to stimulate red blood cell production. But of course, it's a recombinant protein and it has to be injected. And now there are in development drugs that induce the activity of the factors that we identified, the hypoxia-inducible factors that control EPO production, and these drugs can be given as pills by mouth, so that the treatment of anemia can be made much more convenient for patients both with kidney disease and other causes of anemia. And there are 4 different drugs that are all in advanced clinical trials now. Over 25,000 patients are being studied in these trials, and one of the drugs has already been approved for use in China and Japan, and will probably be approved in the U.S. and Europe within the next year or so, if everything goes well. So that'll be the first application of our discoveries to the clinic. Another area, so in that context. The hypoxia-inducible factors are, as I said, play an important role in physiology, and when they're not produced in sufficient amounts, we'd like to stimulate that. But in cancer, the factors are produced at very high amounts because oxygen becomes very limiting in cancers because the cells grow so rapidly that they basically outgrow their blood supply. And become very hypoxic. And this can lead, in fact, to the cancer cells dying because they don't have enough oxygen, which, of course, you'd say, what's bad about that? That's what we like cancer cells to do, to die. But, of course, the cells that are very far away die, but those that are closer, they can survive. But their exposure to low oxygen changes them, makes them more invasive and metastatic, more difficult to kill. And we believe these cells are really cells that are very dangerous in terms of the risk of the cancer spreading through the body and resisting therapy. And pretty much all of the cancer therapies are targeted to the cells around blood vessels that have lots of oxygen that are rapidly dividing, whereas the hypoxic cells are not really targeted by any therapy. And we think that if we can inhibit the activity of the hypoxia-inducible factors in the hypoxic area of the cancers, that will complement the existing therapies and lead to a better outcome for patients with advanced cancer. And so there's a HIF inhibitor that's now in clinical trials for kidney cancer. So I think we've already started down that road as well. When I say we, I mean the scientific community and the pharmaceutical industry, recognizing the importance of this area for targeting in different diseases. Most applied research comes from a foundation of basic research. And what I mean by that is that the basic studies are often done just trying to understand a system and the properties of that system without directly saying we want to target this particular disease, because that tends to be less effective than to say, let's just learn about the system, and by doing so, over time it'll become apparent how we might target the system therapeutically. And so really that's how we started, just with very basic fundamental research. And it's led now to, I think, many applications being apparent that again will take time to unfold, but will hopefully result in new treatments for a number of different diseases.